issue on development. Okay? When you're in this stage, right up to about this level right here, up to about 30 days of development, one of the problems that can happen is that the development of the whole brain and neural system comes from the ectoderm. Right? It has to involute, to fold in, and then connect the upper part to form the neural tube where your spinal cord and brain are going to be forming. So they have the stem cells, the same cells that make your skin, make your nervous system. They come from the same origin, the ectoderm of the embryo. Okay? So that has to fold in again, form a tube, and then come across and close up the top. Okay? If that doesn't close, you're going to get interesting problems. Okay? And they're not too uncommon. Again, if you can imagine, where we start out as this flat embryological plate here. And if you can see here, the top of the ectoderm is coming over. It forms that little indentation. It comes over and then closes over the top there. That is where your spinal cord and brain are going to be developing in the neural tube, right there. Okay, and then the vertebrae have to develop around that. But if this never closes, you're going to get things like this. Okay, this is a relatively minor problem here, where part of the neural tube didn't close, and you get a mild form of spina bifida. Okay. But this can be anywhere from a cyst to a fair number of vertebrae that are open, okay? And depending on the damage and how much stretching of the cord there's been going on, you know, the child may be paraplegic below the level, okay? This is the absolute end of what can happen, where the neural tube didn't close at all. That's spinal cord there, okay? And the brain doesn't develop, okay? These children are, you've probably seen pictures where it looks like they have no head, but they may have eyes, okay? And they'll usually die within a few hours right after birth. This is called anencephaly, okay? Without head, basically, okay? Just PBS, okay? And, but this is a really bad neural tube defect right here. But in case you didn't know what spina bifida is, that's the problem, okay? And this child, this might have, I'm almost positive it was an abortion, because it looks like it might have some saline burns on the skin here, it sort of pickles the skin a little bit. But this is the typical reason what you will see when people are having third trimester abortions. There's almost no other good reason, okay, unless you find something like this and the mother just says, you know, I cannot go on, you know, so, because we know it's fatal, okay. So, yeah, so just so you know, okay. I'm going to go back to, well, this shot. Remember these areas for me: Broca's area, Wernicke's area. We'll talk about that again today. Remember what the homunculus is. It's just you're spreading out the sensation and motor part of the cortex, and this just sort of shows you what. I already did that. Let's go on. Remember the three meninges for me. Okay, the dura, the arachnoid, and the pia. Okay, remember that the cerebrospinal fluid is underneath the arachnoid. Okay, so it's in the subarachnoid space. The falx, very important clinically. Okay, it separates the two halves of the brain. For those of you who've already seen us, you know, dissect the sheep brain, you know that they don't have a falx like this. It doesn't separate the hemispheres. The top part of their brain is connected. The two hemispheres is connected. So, and remember that it also sends down this little ledge on the side, the tentorium cerebelli, and that protects the cerebellum. But clinically, it's important because when the brain starts to swell or is damaged, it's going to be moving along these edges, and things are going to happen. You know, you're going to get brain injury from that kind of thing. So that's the reason I bring it up, why we even talk about it. Okay. Subdural. Okay. As I said, remember the circle of Willis forming, the basilar artery, because it's so important for the pontine vasculature. And then the two vertebral arteries that come up and form that basilar artery. Just be able to point those out if I show you a picture like this. Midline shift, not something I'm really going to test you on, but I think this is something you'll definitely see in your career. Let's see if I can stop this in the middle here. What if I just stop it right there, about the level of the eye? Do you remember the name of the fibers going front to back?
The fornix,
Association fibers,
okay.
yeah. Just the general term, association fibers. And the ones that are going side to side through the corpus callosum,
The fornix.
commissural, right? And then the ones that are going down, up and down the cord, projection, right? Just general terms. Let's catch this in the middle again. Pretty good. This is what that mid-sagittal view of the brains you've been seeing, the models in the lab, and in the book, it has a nice shot of like this. So let's just, again, just briefly go over some of these structures. The whole thing, called the cerebrum, okay, the cortex, and we have the cerebellum. I'll let you guys answer so you get used to it. Cerebellum, right? The little axons, the white matter here, arbor vitae. Arbor vitae, just like vital, it means life. Okay. And arbor means tree. Okay. And a very noticeable white matter tract here.
The fornix.
Corpus callosum.
I want to correct
Corpus
as well.
callosum. And the space.
Fornix. No, third ventricle.
Yeah. Well, it's
That's the septum.
well, there doesn't look like there's a septum here. Okay, I may not have stopped it right in the middle. Otherwise, we would see fibers of the septum. Yeah, it's one of the lateral ventricles
The
here.
third ventricle.
Okay. And you have two of them, and so you could actually count those as 1 and 2. So that's why the third ventricle has got its name.
Okay, so that's not the third.
So this is the third right here. The fluid that comes through the interventricular foramen underneath the fornix there. Well, the choroid plexus there, yeah, that's what's going to be making the cerebrospinal fluid. Okay, it looks like a little ratty vein. Okay, and there's some choroid plexus up in the ventricles as well, and there's a little bit down by the cerebellum here too. But the biggest one you see is usually sitting right on top of the thalamus. Okay, and those are ependymal cells, part of the glial cells in the brain. Okay, so they're making the fluid. And what's this roundish area here? Thalamus, right? And what is the distinction the thalamus has as far as function? Everything, all sensory stuff, right? The sensory stuff that's going out to the cortex goes through the thalamus. Thalamus is important not only for that, but consciousness. Okay, thalamic strokes can put you in a coma because you're losing all that sensory input. Okay. Right below the thalamus, hypothalamus, good. And the little tract here, white matter.
Cerebral aqueduct?
No.
You said a little tract like that?
Yeah, that's a white matter tract going to the eye.
Optic nerve.
Optic nerve, right there. And where it crosses over, right above the pituitary, is the chiasm, right? Okay. And remember, it gets the name chiasm from the letter chi, the Greek word for the X. So that's how it gets the word chiasm. Okay. Right behind that, you're going to see a little stalk holding the pituitary.
Infundibulum.
Infundibulum, good. Okay. And then right behind that, right dorsal to that, we have the mammillary body, right? Right. Okay. In this area right here.
That's
The peduncle.
Well, the peduncles would be close because
The
this is the middle.
midbrain?
This is the midbrain.
The midbrain.
Yeah, but the peduncles are fibers that are splitting off and going into each hemisphere. You're not going to see the peduncle when you split it down the middle. So,
So what was that, the midbrain?
Midbrain, right, or the
Okay.
mesencephalon, call it either one. There's a fluid tract. The third ventricle is going to be shoving fluid down through this little tube right here.
That's the aqueduct.
Right, cerebral aqueduct, or aqueduct of the midbrain, you know, there's a couple names. The roof of that is the corpora quadrigemina, right? Okay. Corpora meaning body, okay, quadrigemina also meaning four bodies as far as I know, okay. Gemina 1, Gemini 2, twins, right? So, and then the upper part is called the superior colliculus. A colliculus means a hill or a bump in Latin, okay. And then the inferior colliculus. So, and we were talking about how the superior colliculus, for our purposes, is mainly involved in visual reflexes. Bright lights flashing, you automatically look at them. Some of your muscle reflexes are related to that as well. And then the inferior colliculus is auditory, right, the hearing reflexes. Okay. Now the fluid goes through there, okay, down into this space.
Fourth ventricle.
Right, exactly. And then it's going to keep on flowing until it goes out into the area around the brain in the subarachnoid
Meninge.
space. It has to pass through the foramen of Magendie or the foramen of Luschka on either side. Okay. And then it's not quite in the middle because I don't see a central canal of the spinal cord here. Okay, but there is a little bit of fluid that goes down the middle of the cord as well. And there are some neurological problems and deficits that can happen when you get that central canal enlarging under too much cerebrospinal fluid pressure. Okay, so. But you can see there's definitely a space right there, that dark space, so that fluid is going down all the way along the cord there. Okay, and let's see what else. Okay, again, we don't really see the falx, but it's there. That might even be part of the fibers of the falx there if we're close enough to the midline. But we definitely see the roof over the cerebellum, and that's the tentorium cerebelli. Right. Okay, so. Think that, well, except for the pineal gland, I think we left that off, okay? And that, like we say, for our purposes, it's enough to realize that that has a lot to do with biorhythms and sleep-wake cycles and things like that. Some of your hormonal cycles might even be tied into it as well. So.
Dr. Ernst, on that picture, do you have the pituitary gland?
It looks like we're either seeing bone of the sella turcica or the pituitary right here.
Oh, okay.
And it's hard for me to tell if that's the actual gland or that's just part of the sphenoid bone.
Okay.
What's this right in here? What do you think?
The sinus.
Yeah, sphenoid sinus.
Venous sinus.
Sphenoid sinus. There's some air in there. How about this muscle back here?
The occipitalis.
Yeah, probably splenius or trapezius.
Back here.
Trapezius. One of the ones that comes up and hooks to that occipital nuchal ridge there. You can see your vertebrae in here. Okay. You can actually see the disc here, these dark spaces, those are the discs. Okay. So if there was a slipped disc that was bulging, we could see that pushing against the cord there. So you can see that MRIs are very helpful for diagnosing a lot of things. I mean, they're actually pretty accurate nowadays. The old ones weren't quite so good, but nowadays they're getting a lot better with the resolution. So,
Is this a healthy MRI? I mean, from what you're looking at?
from what I can see, this is perfectly normal. So, it looks like you might even have a little funnel sinus here. I'm not sure.
Can you, the sausage part of the brain, what was that called again?
The gyrus?
The gyrus.
Yeah. Plural gyri.
Plural gyri.
The little
Okay.
creases, these are all called, one is a
Sulcus.
sulcus, right? Or sulci is the plural.
How would you spell sulci? Just S-U-L-C-I? Okay, thank you.
Okay. Well, let's move on. Now that you've seen what you're more likely to see out in the real world. Now let's look at something like this. This is the kind of picture I'm going to give you. It'll be very similar to actually this one probably. Actually, I think on the test I do use this picture as the mid-sagittal view. And I'm going to have, I've made up my own arrows, okay? So I'm going to be pointing to different things on here, and you just have to label the parts. That's your lab test, okay? And so on here you can see everything that we just went through. And, but as you see, the more you look at this, it doesn't look like a strange land anymore. You can see that these parts and pieces make sense. You can see how they're kind of connected to each other. It's just like learning a little map, because most brains are pretty similar. Okay, even the sheep brain. You can find almost all these structures on the sheep brain because it's a mammal. Okay.
On that, what would be considered the third ventricle?
This space right here. Okay.
There.
Remember, it's pretty thin.
Okay.
Because you have that thalamus and hypothalamus are, from the two halves of the hemispheres, are pretty close to each other. So you're looking at a real thin space. So when I cut the brain in half, I'm looking at the side of the thalamus. And I have to imagine that there's just fluid floating in front of it. Okay?
Okay, so that's
This is a,
And then that goes down
the
that
third
way.
ventricle is a fairly thin slice where the fluid can flow through.
okay.
Okay. So what we're actually looking at is thalamus here and hypothalamus here.
Right.
But obviously in three-dimensional view, they would be going inside the hemisphere. Okay. Their mass is mostly inside, not on the edge there. So, and then we got temporal lobe down here. Okay, pons, medulla, and you remember what the major thing you need to remember about the medulla?
Sorry.
That's where
Movie star.
from the left side to the right side.
It's where,
Where they, I think you said it right.
yeah.
That's where the nerves cross over from the left side to the right
Exactly,
side.
exactly. This is where the majority of motor and your pain receptors cross over. Okay, there are a couple of little exceptions, and we'll talk about those when we get to the cord anatomy.
So the medulla is where they cross over from left to right, but the pons also acts as a bridge, but that's going back to the cerebellum.
Well, not only that. I mean, the nucleus for some of the cranial nerves is in this gray matter in here. So
Okay.
it's not all white matter.
Okay.
The midbrain and some of these darker areas right here have some fairly important functions for, like I say, the nuclei of your cranial nerves, because they're coming off this area right here. Okay? And we'll see those in a little bit. We'll hit them today. So it's not all just fibers. But the motor fibers that are coming through are connected through the cerebral peduncle here with the cerebellum, and that controls a lot of our coordination.
Okay.
Okay? So the basal ganglia, you can barely, well, you really can't even see in this view, because they're deeper. They're inside the hemisphere.
Okay.
And that's why I showed you this picture. Okay, you can see these structures are twisting around here. This is the caudate nucleus and the putamen and the globus pallidus here. This space right here is where the fibers from the motor and sensory strip are going to be running down and then running through this hole and then running down this way. Okay, they're not showing you that, but that's what that space is. That's where all those fibers are coming through. That's the internal capsule.
Okay.
Okay. So when you look at a picture like this is the internal capsule. It's running right through
Okay.
on the inside of the putamen and the globus pallidus.
What type of tissue would that be? Would be,
This one?
no, the
The-
fibers that you're talking about?
Well, fibers are all axons. That's why it looks like white matter. Okay? And but as a whole, this structure right here is called the basal ganglia. Remember what I said that it's kind of responsible for?
I know that if you have a stroke there, it's really bad. I remember I was responsible for it.
Yeah. Let me just, everybody's seen somebody who's drunk. Okay?
Yeah.
Well, picture that as a cerebellar problem. That's coordination. They can't do things, they can't walk, not rightly. Okay. Now, cerebellar disease will do the same thing. I mean, cerebellar pontine degeneration, older folks especially,
will get the same kind of problem where they can't even put a fork to their mouth, they can't eat because it's so uncoordinated. Whereas the basal ganglia have sort of a different classical type of movement disorder that they're associated with, and it's more like tics. It's writhing motions, and you've probably seen some of that too in real life. And the upper extremities can twist and turn and give you weird, kind of motion like that. So it's not a real, well, it is a coordination problem, but a different kind, a different sort of problem.
That's basal ganglia.
Basal ganglia, right. Look, a lot of the deficits from early anoxia at birth, cerebral palsy, are due to basal ganglia disorders. Okay, that's kind of a typical, movement that you'll see with cerebral palsy. Okay, and some of the rapid movements that you might see. Okay, that's called ballism, ballistic meaning striking out, hitting. Okay, hemiballism is the one hand doing that. But you can get it with the feet jumping like that. Just Google or go to YouTube and look at movement disorders or basal ganglia, and you'll see people that have published the kind of movements that you can see. If we have enough time during like a review session, I might pop in a couple of minutes of that, and we'll look at that. But so just associate basal ganglia with movement control and movement disorders when they go bad. Okay. You don't have to worry about the specific kinds of what they're called. And then if we go back to this one, you can see how that might be true, or at least it's reasonable to think that here's the head of the caudate here on a transverse section now. You have to use your 3D powers to see. We're now cutting front to back in the brain, taking the top of the brain off, okay? And we're seeing the front of that caudate nucleus right here, okay? Let's go back. We're cutting it right there, transversely, going front to back. So all I'm going to see is sort of a round piece of it here when I look at it from above, okay? And over here I'll see another piece. And right beside it, this is where the fluid ventricles are, okay? They'll go right down inside there like that, which is why here you have the ventricles, okay? And this is the septum pellucidum. This is the fornix. Septum is attached to the fornix here. You see the head of the caudate, okay? And if you're really good, if it goes back here, it curves around, you're going to see the tail of it, which I believe is right here. Okay. And that's the other part of it as it curves and down around. So, but they're right next to the motor and sensory pathways. So, and because they're gray matter, these are neurons. Okay, so this is going to have inhibitory or excitatory function on those motor neurons to either stop weird movements or to initiate them. Okay, so. But internal capsule, just burn that word in your head because that's important. That's that projection pathway that's going from your motor center out down to the brainstem, and the sensory stuff is coming up in the same place. Okay, so there's motor and sensory here in the internal capsule going up to the cortex. Okay. I've been asked, where's the insula? Well, this is the insula right here. It's the lobe right inside the temporal lobe. If I were to take that temporal lobe and sort of pull it out, you look down in there and you can see the insula. Okay. You can't do that on a sheep brain. It's not built like that. Sorry? Okay. What else is here? Well, you see we cut just at the level of the corpora quadrigemina. Okay. And if we cut a little lower, it looks like this. We cut off the superior colliculus. So this is just a different level, okay? This is a little higher, this is a little lower. Okay? And so it's going to have just a slightly different appearance. The caudate is going to be a different size, and that sort of thing. You can actually see the fornix is starting to split here. Okay? Because what you see in the midline is just that fiber that's running in the middle, but the tail of the fornix splits and runs around just like the caudate. And the head also splits and runs down to both mammillary bodies. Okay. So, what would this fluid space right here be?
That's the fornix. That'd
Good.
be the
Third ventricle, right. And that's between the two thalamus, or the two thalami, I should say. Okay.
So you have two down.
So, okay, so caudate, putamen, globus pallidus, internal capsule. Okay. This is a similar sort of thing, but you're looking now at a coronal view. Okay, now we've split the brain this way. Okay, so the caudate will be running this way. Okay, and we have just now taken a slice of it. Okay, so there it is again, right by the ventricle. Okay, again the septum pellucidum. Here's the corpus callosum, right above it. And we're looking at part of the roof of the corpus callosum now. We're not looking at the front part. We're not looking at the genu when we get a transverse section. Okay, now we're cut down through the middle of it. And we're looking at the what you might call the roof of it.
And that's both sides?
It's both sides, yeah. Yeah, here's the axons. These are the commissural fibers going from side to side. And this is really how your brain talks to itself, okay? These fibers communicate from right to left. You read sometime about syndromes that happen when you cut the corpus callosum, and tests you can do on people like that they can't communicate, their brains can't communicate with either side. And sometimes you can, if you block their vision, okay, remember the left side can talk, right? The right side doesn't have a verbal center. So you can show the right side something in a certain visual field so that the fibers, won't cross, because half of the fibers in your visual fields cross over, okay? But if you show somebody in the field that's not crossing over, they'll see it, and they might laugh if it's funny. Like, somebody jumping up and down on a table naked or something, that, they'll go, ah, and then you ask them what they saw, and they go, I don't know. So it's weird that side of the brain can't talk, but it's there. And I've heard some people say that they even have different personalities. So, and so yeah, it would be interesting to read more on that. I'd like to too. I might do that this summer. But here again, the putamen and the globus pallidus, the internal capsule. These are the fibers coming down again. Okay. And the reason this is called the internal capsule is because this part right here is called the external capsule. Okay. And then there's a structure right here. This gray matter is called the claustrum. Okay. And then you have the extreme capsule out here, but those aren't on your list, so don't worry about it. So, you're getting down into the hippocampal area down in here. This is where most of your memories are forming. When you form memories in the present. Okay. So when you have damage to the hippocampal areas, you have a very poor memory recall. I think I told you about the fellow that had both of them removed surgically. There's a famous case. They think he's probably been one of the most studied human beings ever. Back in the 50s, and he actually just died in 2008, I read recently. But for seizure disorder, they took out his hippocampus, and they had no idea that they were going to deprive him of any further memory formation. So he literally could talk to you, walk out of the room, come back in, and act like he'd never met you before. Okay? Because he couldn't form any memories. He always lived in the present, and if he had to do something, he had to write things down so that he would remember them. Okay, like to-do list every day. Okay, so it was a really sad kind of case, but they called him HM, I think, for his initials. Okay, I don't know his real name, but sad case. And, all right, what lobe are we talking about here, though?
Parietal.
Temple, oh,
Occipital.
No, temporal.
Temporal, yeah.
Temporal.
Remember, this is a coronal view.
View. So,
You're cutting right down through the middle here, so the side is going to be temporal. Okay. So yeah, that's not occipital back here. Okay, this is a coronal view. We'll go back one. Now you've got occipital, right? This is the transverse. Okay, temporal is out here now. Okay, but now we're going to cut it this way. Okay, and now that's what you.
but it looks so similar.
Very similar because these things are curving.
Okay.
Okay? So sometimes they look similar when you cut them that way or that way.
Right.
And that's why the caudate looks like it's in the same place.
Okay.
So, well, maybe one more time, let's look at this. See, if we cut it this way, you're going to see two caudates with the ventricles right there. Okay. If we cut it this way, you're going to see two caudates with the ventricles. Okay. That's why it looks similar.
Okay.
And just got another one that's sort of labeled for you,
Oh,
another
yeah.
coronal section. So this would be temporal lobe
Yeah.
right here.
Yeah.
Insula in here, okay, behind the temporal lobe. Now, this would be parietal. Okay.
Right on.
Okay. And, okay, all right, I think you've got a feeling. Look how much the fornix is split here. Okay, it's no longer just that simple midline structure, it's starting to split. Okay. In the sheep's brain, the fornix is much wider than ours, so you will see what looks like more of a split. Okay. All right. And have we gone over this yet? Can we get this slide?
No.
No? Okay, let's talk about it there. Just to be sure you know the path of that cerebrospinal fluid again. That's all this is for. Here's your choroid plexus. You've got some of that tissue here. You've got a little bit down in here as well, close to the fourth ventricle. But most of it's going to be made up in the ventricles, and lateral ventricles, and the third ventricle. It's going to pass down through the cerebral aqueduct here, okay, into the fourth ventricle, and then eventually it's going to work its way outside the tissues in those foramen of Magendie, foramen of Luschka, okay. Magendie's in the middle, Luschka's lateral. And it's going to flow around in that subarachnoid space. It's going to go up and over the tentorium there, and then flow around the brain. Okay? And same way around the spinal cord. This is why you can do a tap down here and get spinal fluid. Okay? So if you want to know what's going on in the brain, if there's any bacteria in the fluid, if you got meningitis, that way you can tap it here and you can culture the bacteria. Okay? Or if there's been any bleeding, you're going to get a blade tap too. Okay? When we talked about the meninges, we already talked a little bit about the arachnoid villus or granulation, how you get the purifying effect when the toxins are put out into the venous blood out here in the sagittal sinus. That comes down right here where the tentorium splits up like this. This is where that jugular groove is that you saw on the bones. Okay? Then it's going to flow off to the sides, which you can't see here, and go down the jugular foramen and out the head. So you don't really have, the biggest venous pathways aren't really veins, they're connections of this dura. Okay? So, all right, so what can happen when things go wrong? Well, remember that your eyes develop as part of your brain. They really are truly brain tissue. They bud off the diencephalon and come out. And so there is dura covering your optic nerve, okay, as it goes to your eyes. And then that seems to stop, but the rest of the eyeball is actually made of tissue that's very much like the dura. The hard white of your eye, that hard part, is the same kind of tissue as dura. Okay? And so what can happen is that if you increase the cerebrospinal fluid pressure for any reason, you'll build up the fluid pressure underneath the dura there, and it will cause pressure on the back of the eye. Okay? This is a normal eye. This is the optic disc. When you look at it with the ophthalmoscope, you'll be able to see this in the lab next week. I'll show you how to use those. This is what a normal eye looks like in the back. The optic nerve this is where it comes into the eye, and there's no vision right there, okay, because it's all nerves. There's no photoreceptors. But this is where this optic nerve is right here, called the optic disc. And when you put a lot of pressure, fluid pressure behind it, it starts to swell and get edematous. So when you see something like this, you got to worry about increased pressure in the head for whatever reason, okay, like tumors and things like that. So, and people who come in to the doctor to get checked out for headaches and things, he's typically going to look in your eyes to make sure you don't have this, okay, because that's one thing you don't want to miss. I've only seen it a couple of times in practice, but you certainly don't want to miss it, especially if somebody was complaining about headaches, which would be a typical cause of increased cerebrospinal fluid pressure, right, for tumors and things like that.
I have a question.
Yeah.
Some people have discovered eye tumors or brain tumors with pictures. Like when you take a picture and your eye reflects something. I don't know if you ever heard about it.
Well, I think what you're talking about is melanomas. If you have a melanoma on the back of the eye, and I'll show you a picture of that next week, you don't get the red light reflex because it's a dark tumor.
But you get the shiny, so
While you might not, you'll just see reflection off the cornea. But you don't get that pinkish reflection from the retina on the back of the eye if it's dark. So,
Or other growth, any kind of growth in the eye. Any kind of,
Just like,
it will cause that reflex to go bad. But you're also not going to get it with cataracts. I mean, the light can't even get through and get out. But this is called papilledema. Okay, I'm not going to hold you to that, but that's something, a word you should hear. Okay, papilledema. So now, what else can go bad? Really bad. Well, one of the things that can happen either congenitally or because of tumors and injury, you can block the cerebral aqueduct, and now the fluid has nowhere to go but build up in the ventricles. Okay, and that's what causes this. Okay, this is a young child with hydrocephalus, literally meaning water head or water brain. Okay, you can see those ventricles are huge, and they're just pushing brain tissue out. His sutures haven't closed. Okay, so as the brain gets bigger, the head just keeps growing. Okay, you basically never close those fontanelles.
But they can drain that fluid out?
Oh yeah, you're getting ahead of me here. This is
It's
you don't
so
see
sad.
this too much anymore unless the child has just been neglected. Okay, because you can go in and you put basically a catheter, a shunt, in the ventricle. Okay, and then just run it down inside the neck and the body, and then let the fluid drain into the abdomen. Okay, and then it's reabsorbed. Okay, they'll even put little pumps on the catheter so you can pump out fluid if you have to. And then as you get older, you have to change and get longer catheters and things like that. But you almost never see this in a first world country anymore because of that. You can't fix this, and that's why pediatricians are checking the child's head every time they're doing head circumference and height and weight, things like that. You don't want to see that head circumference start to grow off the chart. Okay, because then you'd have to be suspicious of something like this. If the child didn't already have some neurological issues. Okay? But this child is, looks like they're not abused, but they're going to get bed sores on their head. That's what that is. Because he can only lay in a couple of ways. Okay? So you're going to get bed sores just like somebody who can't move. Okay? Yeah, poor little guy. But, I just thought I'd show you what a brain tumor looks like. Okay. I'm not an expert on these, but my impression is this was a fairly slow growing tumor because there's not a lot of swelling around the tissues and bleeding and that sort of thing. So, but they can have the consistency of sort of like cheddar cheese. That's the consistency of a lot of these tumors. Okay. But you can see that this was probably fatal. Okay.
And is that, if I'm looking at it correctly, the frontal lobe?
That looks like frontal lobe, yeah.
Okay.
Yeah, because there's no other structures down here. So we're in front of the caudate and we're in front of the corpus callosum, so you don't see them. Okay. So we're taking a slice through the frontal lobe.
Does that tumor essentially just eat up all those axons and nerve bodies and all that?
Yeah, but not always though. Sometimes it might even be encased almost in a sheath, and it's just an enlarging ball, and
Okay.
squeezing everything else around it, pushing it out of the way.
Would that be inoperable?
Well, apparently.
Yeah, for this particular.
But sometimes they are inoperable, yeah. But the more encased they are, the more operable they are, I would think. Okay.
Yeah.
So, but yeah, if you've got a real one that's just got fingers growing all through the brain, that's inoperable. This is a lacunar infarct, usually caused by high blood pressure. Why do they call that a lacuna? It's a hole. A lacuna means hole, right? Okay. So infarct that would be more to your point. The infarct, anything you infarct, like a kidney or a heart or whatever, you cut off the blood supply to something that's called an infarct. So a myocardial infarction is a heart attack. A renal infarction is basically a stroke in your kidney. Okay. This is a cerebral infarction. This is a pontine infarction. Okay. This is the pons. But it's called a lacunar infarct because it leaves these little holes behind after it heals up. Okay. This may not have killed this patient. I don't know if this was the fatal cause, but certainly big ones can. Okay. And that's that basilar artery that's responsible for giving you the blood flow to these areas.
How did you know that was a pons? Because it's kidney bean shape, or just because you just know?
Well, basically, there's not much in the brain that's round like this, other
Okay.
than the pons or the medulla,
Okay.
you know, and it's on the bottom.
Okay.
You know, it's on the ventral side here, and you got the artery. So I knew it was midline.
Okay.
There's really not much else in the midline that looks like this. So, but these things can be bigger depending on the size of the stroke.
What is it, like jelly or something?
It just sort of liquefies, and then the microglial cells will clean it up and reabsorb it, and it leaves you a little hole like that. And you could have more than one, you know, you could have them all over the place.
And you can still live with it?
Depending on the deficit,
Oh,
yeah,
yeah.
yeah. A big one gives you what they call a locked-in syndrome. Okay? You basically, if you get a big pontine stroke or lesion, you basically decapitated your head from the rest of your body. It can no longer communicate with the rest of your body. The only thing, if you live through it, the only thing you can do is blink your eyes.
But you can still think and
Oh yeah, your
and
communication is perfectly normal.
you can still see and
Just
hear,
imagine
wouldn't you?
yourself lying in bed. The only way you can communicate with the world is blink your eyes. That is horrible. It's horrible. I mean, I've seen 2 cases. It's really tragic.
Can they feel anything?
No, they're totally cut off.
Is-
Motor and sensory.
Is that the same thing as the Terry Schiavo, or is that something different?
No, she had brain injury.
She had brain
She
injury.
had a third of her brain that just atrophied from anoxia.
Okay.
So she really didn't have much cerebrum at all to think with.
Okay.
Okay. But this is a little different. This is cutting off the highway that's controlling all the messages going from top to bottom. Okay.
So
Which,
would TIAs be associated with that?
no, a TIA by definition is a reversible sort of thing. So if you had, let's say, a transient blockage in a small arteriole here, and it affected the passage of the signals through this area, and then the blockage went away and nothing died and it all healed up, that would be considered a TIA, transient ischemic attack. But they're not usually medullary or pontine, they're usually cortex. Yeah, so it's usually part of the cortex. Okay. And here's a big cerebellar cerebral infarct. You can see that it's almost starting to liquefy here. It's obviously it's an autopsy study, okay? So this patient didn't make it. And occasionally you'll get other infarctions too. Okay? You'll see multi-infarcts. Sometimes people don't even know they've had a couple. And they come in with some sort of complaint. They might not even know that their right grip is a little weaker than their left. You know, they might be complaining about a headache or something, but they're not even aware that they've already had a stroke that's affecting them. Okay?
Is that a coronal view as well?
That's coronal, right? Right. You're looking from the top down, so these would be your temporal lobe again. Here's the insula.
Okay.
The fornix is split, okay, and here's your thalamus on either side of the third ventricle.
Okay.
But then here's the internal capsule.
Okay.
Okay, this is where the fibers are running down. You get a good view of it here, running down. They'll be going right down into the brainstem.
Okay.
So, but they're coming from the motor strip and the sensory strip up here. Okay, so you've had a big wedge, kind of a major artery here that it's just, you know, it's like a hand. You can cut off a finger and get a little stroke.
Right.
But if you cut off the wrist, you get everything.
Right.
Okay, and so you get this big wedge of tissue. Okay,
Right.
and that's what you'll see. Okay. Here's an interesting issue. AVM, arteriovenous malformations. Many of these are congenital. Okay, they may get bigger as you get older, but it's basically a messed up conglomeration of little arteries and veins that wrap around each other. Sometimes there's even a shunt between the two, so that the artery is pumping blood into the vein. Okay, but they tend to blow up like a balloon. Okay, and then they just rupture and burst. Not too uncommon. Not too uncommon. There's a real nice surgeon in town here that had this while he was operating. Okay, and he never practiced again. So, but you can see the big clot there where the original vein was, I think. This is just bleeding into the ventricles. This is all blackish, clotted blood. Okay, so, but you can see that it was also bleeding into the tissue of the brain, and that's not good. Okay, so obviously an autopsy shot there too. Okay.
I think we've already talked about Wernicke's and Broca's aphasia and those problems, so I'm not going to spend a lot of time on this right now. But remember, the motor area is in front, the precentral gyrus here, the motor cortex. Associated with that is Broca's area, which is the motor for speech. You knock that out, you really have a hard time getting any words out at all, even though you understand language, you understand what you want to say, okay? So, and then, oh, I'm getting them backwards here. I'm talking about Broca's, but the slide is for Wernicke's, sorry. But on Wernicke's, it's just the opposite. It's a sensory problem, okay? You don't understand language. You don't understand what people are saying to you. You may be aware that you're talking, but people who have recovered from this say that I didn't know what I was saying, okay? Because the grammar and understanding of language is gone, okay? But if you survive to report about it, I'm sure you'd have some interesting stories, okay? It's just gibberish. Many times they talk a lot, too. They don't even want to stop talking. So I called my mother on the television, okay? All right, it just doesn't make any sense. And then Broca's, again, is the opposite. You might get out a few words like this to make yourself understood. Son, university, smart, boy, good, things like that. If it's worse, you may not even get that out. You may not even get more than somebody says, are you hungry? You can understand the language, and all you can do is and shake your head. So that's very frustrating for people. And they very often get treated like little kids, and they don't like that very much. Okay? So.
But they could write down. They still have the ability to write down.
Visually,
They just can't spit it out, basically.
Yeah.
So it's constantly
But
gives
there
something
are-
to tip your tongue.
There are expressive aphasias, though, like, I think it's more associated with Wernicke's, though. There is agraphia, where you lose the ability to write and read, too.
Okay,
Okay, I think that's back in here.
back in there.
Back in
So it'd be
So
like
you
you.
would just lose all forms of communication if you
Pretty
had a
much,
problem
yeah.
there.
Because you no longer understand the language.
Anything,
Yeah, so you can't write.
My cousin, when we were younger, he had a lot of, I forgot. Seizures.
Yeah.
And every time he would have the seizure, everything he had learned, he would forget it for, like, he had to start all over.
Learn
Like,
what?
he couldn't write, he couldn't,
Oh,
he
interesting.
couldn't hardly speak. He had to almost learn everything all over every time he had a seizure.
Well, that's interesting. I haven't heard of that kind of memory loss with seizures.
I was wondering if it would have something to do with one of those areas.
So how would you explain, knowing that my daughter has Down syndrome, how would you explain she can walk, she can eat, she can, use the bathroom, but yet her speech is delayed, or her articulation, is the whole brain affected by a syndrome?
Well,
Is it the whole brain like compromised?
In that case, yeah.
It's just so weird that she can do everything other children do, but yet then she has her slow parts. And where
Yeah.
during development does that just pinpoint those areas, such as,
Well, your guess is as good as mine as when and how it happens, because I'm not even aware why an extra chromosome would even cause Down syndrome.
right,
And I'm not sure anybody really can explain it.
right. They just know it exists.
Yeah, exactly. But you're talking sort of global deficits with cognition.
Yes.
Okay, and speech and motor development sometimes, where they can be uncoordinated and
Right.
that sort of thing.
Right.
So you're talking a global brain problem.
Right.
Okay, and if they get normal at anything, it's because of training and plasticity of the brain, learning different movements and things like that. But these are, Yeah, so that's different, so that's a global problem.
Yeah, yeah.
Whereas these are modular problems, we're talking about strokes in small
Stroke, right,
areas.
specific injuries
Yeah,
to that,
yeah.
whatever causes it, yeah.
And it's, I mean, it is fascinating because, you know, strokes in certain areas, I think we've already talked about how you can lose the ability to recognize faces and even loved ones, and you're no longer, sometimes you're no longer,
Disassociate with.
yeah, you disassociate them from your pleasure center, and you're convinced that that's not really my wife, you know.
It's an alien abduction.
Right, right.
Replacement.
It looks like her, but because they don't feel the connection, they won't believe that it's really her, that sort of thing. And little tiny things as far as like naming objects and knowing how to use them, things like that. You can have little strokes and all of a sudden you forget what this thing is called.
Yeah.
You know, is that a book? I don't know. You know, that's weird. It's called anomia. Okay, so just remember the basics of Broca's and Wernicke's aphasia. And we'll spend the next 20 minutes talking about cranial nerves here. Obviously, it would be tough to memorize where all these things are, okay? So you don't have to, okay? The only two I want you to know for a test, if I show you a picture like this, you should be able to pick out the olfactory bulb and tract for the sense of smell, okay? And you'll see that on the sheet. It sticks out like a sore thumb, okay, on the sheet brain. And then the optic nerve, the optic chiasm, okay, here, the crossover is called the chiasm. You should be able to pick those two out. I'm not going to make you try to memorize any of the others, okay? And you probably won't even be able to find them on the sheet brain, okay? Because as they take the brain out, these things get ripped and torn and that sort of thing.
Where's the
How do you spell chiasm?
C-H,
It's
is
where
it up here?
crosses.
No, it's not.
C-H-I-A-S, chiasm, okay? And I think I just said that that word comes from the Greek letter chi, the X, okay? And, okay, but now, this doesn't really give you all the names and the numbers, but I want you to know them, you'll have to know them by name and by number, so you'll have to know them in order, okay? One of the things that helps, you can't see it right here, there's a little mnemonic, the old clean one anyway, okay? Here's the starting letters of the 12 cranial nerves, okay? And it's on old Olympus towering tops, a Finn and German viewed some hops. That's the clean one. Okay?
Tell us the dirty one.
Turn it
All right,
off.
turn it off.